Welcome to Web Microbiology, a podcast for Bio 204 at Allegheny College of Maryland. I'm Professor Kelly. Let's get started. Episode 8, Laboratory Real Talk. Welcome back. I've been hinting at the differences between how we are learning unknown identification using phenotype and how a clinical lab really operates. In this episode, I'm going to get into those differences along with the modern operation in most labs and how these processes save time, which means more patient lives saved. One of the challenges of clinical micro is that many specimens come from parts of the body that are not at all sterile. The throat, the skin, the GI tract especially, and even portions of the respiratory tract contain large populations of our normal microbiome. Think back to the Ubiquity and Normal Flora labs where you sampled your own body. Most of the organisms you cultured were not causing disease. They were simply part of your microbiome. When a throat swab or a stool sample arrives in the lab technicians are often looking for a potential pathogen hidden among thousands or millions of harmless microorganisms. So to help separate pathogens from the normal microbiota, specimens are inoculated onto selective and differential media. These media suppress some organisms while encouraging the growth of others, making it easier to detect bacteria that may be responsible for disease. At the same time, the context matters. Some bacteria are considered normal in one area of the body, but problematic in another. E. coli is a common and beneficial member of the intestinal microbiome, but it causes UTIs in the bladder. Other bacteria that are expected in a stool specimen would be highly concerning if recovered from blood, for example, or cerebrospinal fluid, which are normally sterile sites. Some infections are also opportunistic, and some only cause problems in large numbers. So for all of these reasons, identifying an organism isn't always as straightforward as it could be. When the quantity of bacteria present is just as important as the type of bacteria recovered, counts become necessary. Urine cultures are a good example. Because small numbers of bacteria can be introduced during specimen collection, labs use calibrated loops that deliver a known volume of urine onto culture media. After incubation, the number of colonies can be used to estimate the concentration of bacteria in the original specimen. And this is just another version of the serial dilution and plate count experiment you performed. A high bacterial count combined with a likely urinary pathogen provides stronger evidence of a true infection than the presence of just one colony-forming unit alone. This allows microbiologists to distinguish between contamination, colonization, and actual infection. Blood agar is commonly used in clinical labs because it supports the growth of many organisms and also reveals hemolysis patterns. Some bacteria produce toxins that damage red blood cells in the agar, creating characteristic zones around their colonies. For example, Streptococcus pyogenes, a common cause of strep throat, produces complete or beta hemolysis, and that leaves a clear zone surrounding each colony. Other Streptococci species may produce partial or alpha hemolysis, which creates this greenish discoloration, and some produce no hemolysis at all. We call that gamma. By examining the patterns, the microbiologist can narrow the list and choose which colonies should undergo additional testing. Again, this helps with selecting for the pathogens. Rather than analyzing every single bacterium growing on the plate, we can focus our attention on the colonies whose appearance is consistent with known pathogens. Although the gram stain is one of the most valuable tools in micro, it is not appropriate for every organism. Remember, there are a few bacteria, such as our Mycobacterium species, that have waxy cell walls that don't really gram stain, and instead they require acid-fast staining techniques. Other pathogens might be too small to visualize clearly with regular staining methods. They might stain poorly, or they might require specialized techniques. So as a result, microbiologists have to select staining methods based on the characteristics of the suspected organism and the clinical history, rather than relying on a single approach for every specimen. So a suspected tuberculosis case might prompt an acid-fast stain first to rule in or out this serious pathogen. Now, historically, this phenotype approach formed the foundation of clinical microbiology. And for decades, labs relied heavily on all the methods you've learned so far. Even today, they do remain important components of identification workflows. However, traditional methods have limitations. Cultures require 1 or more days to grow. Some organisms, as you should remember, are difficult to cultivate. And in critically ill patients, waiting several days for identification can delay treatment and result in poor outcomes. These limitations help drive the development of newer diagnostic techniques. Once isolated colonies have been obtained, many labs use commercial identification panels like the API system to speed up the identification process. So instead of doing dozens of individual biochemical tests in separate tubes or plates or with all the lab equipment you've used, the microbiologist inoculates a single strip containing a panel of miniature tests. Every little chamber evaluates a different metabolic capability, like carb fermentation or enzyme production or even substrate utilization. After incubation, the pattern of positive and negative reactions creates a unique biochemical profile that's compared to the database of known organisms. So these panel systems dramatically reduce the amount of labor required, they standardize the testing procedure, and they provide organism identification within hours instead of days. So this allows the clinician then to make treatment decisions more quickly. If you look in your textbook at figure 18.4, you'll see an example of an API test panel. Now notice the lactobacillus label in the upper right corner. Lab techs can't just pick a panel willy-nilly. They're chosen based on the initial information from those first steps. So the API 50 CHL panel in this figure is used specifically to identify lactobacilli and other lactic acid bacteria. And there are different panels for Staphylococcus species, Streptococcus and Enterococcus species, gram-negative enteric rods, and many others. It's kind of like a dichotomous key on a card. This dramatically speeds up identification, and there are even more newer and more advanced systems also in use, depending on the resources of the lab. Another point to keep in mind is the lab work isn't necessarily complete once identification is made. Clinical labs also need to identify effective antimicrobial agents for treatment. Patients with sepsis or other serious infections caused by a still unknown bacterium are often started on broad-spectrum antibiotics immediately. However, once the pathogen has been identified, therapy should be narrowed to the most effective targeted antibiotic. And labs accomplish this through antimicrobial susceptibility testing. In the Kirby-Bauer disc diffusion method, for example, paper discs containing different antibiotics are placed on a bacterial lawn. And then these zones of inhibition are measured after incubation. Other methods can also be used, like broth dilution, and there are automated systems for this, and they all determine the minimum inhibitory concentration, which is the lowest concentration of an antimicrobial that prevents visible growth. The results help clinicians select the most effective therapy for the specific pathogen. And this approach is known as antimicrobial stewardship. It helps improve patient outcomes while reducing the development of antibiotic resistance. Now, some labs, especially in teaching hospitals, are also performing other related testing like phage typing to provide the most up-to-date therapeutics. Phenotype methods are not the only tools available for microbial identification, and in the next episode, I'll be introducing genetic and immunological methods of ID.